Hello, and welcome to this digital poster for the ISMRM23 conference. This poster's topic is on the longitudinal stability of brain and spinal cord quantitative MRI measures. This work was done as part of the Cooltwell project on neuronal modeling. It involved scanning on one single 3D scanner, 6 participants, on up to 10 time points over the course of 3 years. We investigated several quantitative measures, including T1, MTsat, MTR, diffusion measures, and some morphological quantitative measures in the spinal cord. We used automated pipelines that we developed and shared. For the brain analysis pipeline, the quantitative measures T1, MTR, and MTsat, we used the QMRLab software with a pipeline managed by Nextflow and mediated by Docker containers. For the brain diffusion analysis pipeline, we use the TractoFlow software, which is a pipeline managed by Nextflow and mediated by Docker as well. In this diagram, the flow of the analysis is demonstrated. For the spinal cord data, we used SCT, the Spinal Cord Toolbox, which is powered by multiple open source libraries such as ANTS, DyPy, NiBabble, NumPy, and SciPy. Here are some of the results. For the brain, we looked at whole brain white matter and gray matter mean metrics. Each figure shows the quantitative measures. Each data point is one time point scanned for each subject. And each figure we show the coefficient of variation intra-subject and inter-subject. Here are the brain diffusion results. For diffusion, we looked at the mean values in the genu, body, and splenium of the corpus callosum. In this slide, we show the spinal cord results for the morphological metric, the cross-sectional area. This was measured using 3 different measures. So we measured the spine's cross-sectional area using the T1 weighted image, the spine CSA using the T2 weighted images, and using the T2 star weighted image, we measured the mean gray matter CSA. Here we show the quantitative metrics in the spinal cord T1, MTR, MTsat, and diffusion in the white matter portion of the spinal cord. Overall, we found that brain T1 measured using MP2 RAGE, MTR, and diffusion demonstrated the best longitudinal stability. The reported intra-subject coefficient of variation means indicate good stability of all quantitative metrics measured in the brain, less than 2.3% in white matter and 3.1% in gray matter, throughout 10 sessions acquired over 3 years. For the spine, the CSA demonstrated better stability than T1, MTR, MTsat, and diffusion. We found that there is good long-term stability of the CSA quantitative metric in the spinal cord, and that T2 weighted images is the better choice for CSA quantification. Please note that this is the first anatomical data release of the ongoing Cooltwell Neuromod project, and data is continually being acquired. Requests for datasets can be made on our website. This long-term database of quantitative MRI metrics may be valuable information to incorporate in deep learning training models of other longitudinal measurements such as fMRI or MEG to account for confounding changes in the brains of these subjects. We've also made available an interactive preprint of our manuscript in preparation, which can be viewed on this website, where all of our figures are also plotly figures, and you can look at each individual metrics that were acquired for each session by hovering on the cursor. So please check out this website, and thank you for listening.